Hi, this is Matt with Algae Research and Supply, and today we are going to go over the unboxing of our marine microalgae isolation kit. This is the kit right here. But before we even go into any of those things, I wanted to go over a little bit of the workflow about what we're going to see with this, so that as we unbox the things, it's going to make a little more sense. I'm old, I take my glasses off. There's going to be some microalgae samples that are inside here. A number of them, you're going to, as the instructor, going to blend them all up so you can make mystery samples of it. Then you're going to plate them, have your students streak them, let them incubate for about a month, and then it's going to show up something like this, where you're going to have individual isolate colonies that are growing, and then you'll be able to isolate them and hopefully be able to create your own individual culture with that. All right, so let's go through. That's kind of the goal with the kit. The kit overview is right there too. What's in the kit? We're going to have 3 bags as we unbox them. So let's get into bag 1, bag 2, and bag 3. Savannah is here with me. Why don't you pull out bag 1?
You want the incubator first?
Yeah, let's do incubator. Algae incubator is the first one up there. This one is a 5-volt USB powered incubator. Thank you. Inside of this, we have our seawater agar right here. Microwave this in like 15 to 30 second bursts. It's going to take a little while to get it to soften up. We have a video on that. We'll put the link down there. But this is going to be what you're going to need to make the agar. 20 plates, and then the benchtop light. This is a benchtop algae mat. So we figured out we put these into the takeout boxes, and they work really well to be able to just lay down there. They can get wet. They're not submersible per se. They probably are okay for a short period of time, but operating underwater is probably not a good thing. But this is great because it keeps everything in the light, and it also keeps it out of your way, and you can stack them. Okay, so that's the incubator, 20 petri dishes, the marine agar, and then bag 1. Okay, bag 1. Why don't we have you get him ready? Yeah, I don't want to rip this stuff too, because somebody's getting this bag. In bag 1, we have 5 dense, unique 50 milliliter algae cultures, salts and nutrients to make up the seawater. There we go. 20 sample tubes that are 15 mils, and this is going to create the dilutions. That's right there. That's for doing the dilutions. And finally, there should be 5 sterile pipettes. Those pipettes will never have babies because they're sterile. Sorry, dad jokes are free. All right, bag 2. That's bag 2 right there. All right, bag 2. Here we go. Inside this, there'll be 2 mil tubes of nutrients to add to the agar. So this is going to be these agar bits, and you'll see them in a second. That's going to be added to this, and that's just to give a little bit of food for the algae. There's no sugars in there for bacteria or fungus. 5 1 mil sterile pipetters. We have pipetters in this bag as well. 20 individually wrapped toothpicks. And then finally some parafilm. This is to help seal the plates, so that way they stay nice and clean. You're going to need to cut strips of that. We started to cut the strips for it, but we realized that you and your students will have more fun doing that. So, and parafilm is very fun to play with. Don't tell your students that though. Finally, bag 3. There we go. In this, we're going to have 5 50 mil centrifuge tubes. There we go. Here are the centrifuge tubes. These are going to be to isolate and scale up the algae cultures after you pick them off the plates. 5 individually wrapped toothpicks. And those are in the bottom there. Some toothpicks. 5 beaker bags, which we saw just a second. 5 of the beaker bags. 5 seshi sticks. These are to measure optical density once you get them into the algae culture. That's a way to measure the biomass of the algae that's there and track its growth. 5 seshi sticks, some seawater packs, and then some stuff for the nutrients to actually grow the liquid culture. That's everything that's inside the kit. So again, to start out, and now we're on to the second page of the workflow here. Now that we've got everything as a big mess, that's okay. the first step here is that the instructor is going to make down the culture. So we came up with a kind of just a back of the envelope way to describe mixing out these cultures. The names of the cultures there, pour a little bit into each one of those things there into the 15 mil tubes. And that makes it blend so that you only have 3 different strains in each blend. Some of the strains are motile, some of them are not motile, some of them are brown, red, or green. And so you're going to get a really cool way to kind of mix that up so your students aren't going to get the same thing with each person. Next, it's going to be plating the agar. So that's getting that stuff done. So next, it's going to be melting this stuff down. So that's going to be, again, in the microwave, 15 to 30 second bursts, depending on how strong your microwave is. Uncap it. Like I said, we've got a video on that. It may take 2 to 6 minutes, maybe even 8 or 9 minutes to get it to be liquid. Shake it up once you're getting everything good, and then it'll go into liquid form. You want to make sure that it all melts down. And then just simply pour them into the plates. Do this in as a sterile area as you possibly can. Put a little bleach down. Use aseptic technique. You'll have to dig back into your undergrad microbiology class to remember how to do all that stuff, unless you already teach it, and then you're ahead of the curve. Okay, pour your plates. Set up your lab stations. You'll know how to do that. It's just kind of how you want to organize everything there. Do your serial dilutions. This is kind of a cool thing, so you can practice doing 10-to-1 serial dilutions. Then transfer the algae cultures onto the agar plates. This will help you to do, or this will be a way for you to get the streaks on it, so you can do the dense streak, the medium streak, and then the low density streak. And finally, incubate. Just store them in the incubator here for probably about a month is going to be a good time. Your students are getting anxious about it, want to see if there's any growth on it, so let them. It should be really fun to watch, but try to keep the actual petri dishes closed especially during the first few weeks of the growth period, because even though we don't have any sugars for the fungus and bacteria to grow, they'll grow first and use up whatever nutrients they can get from the agar or just that happen to be there. So they're going to bloom and bust first, but the algae is going to be what grows over the long term. So if you wait it out, there's going to be a lot more algae biomass on there, and just keep it closed. Probably a good idea anyway. So finally, once you get everything growing and you get algae on there, you can pick up the individual colonies, and then get those to grow in the 50 mil containers. And then ultimately, once you get it to scale up, we've got lots of salts and nutrients for you to make down your own media. We recommend using the 500 milliliter bottles of water, and that should be just about it. Just scale it up, and then you can describe it, and then actually even track its growth with the Seshi stick. It's very fun. Disposal, all of these are non-invasive, so you can put them right down the toilet. If you're worried about doing anything of that, just put a little bit of chlorine bleach in there, and that'll kill everything as well. Other than that, you can feed the algae cultures to your coral aquariums or to your brine shrimp if you've got those around. And that's pretty much the kit. So if you have any questions, reach out to us here at Algae Research Supply. This is Matt and Savannah. Hi. And we're here to help you guys out. We want you guys to be able to say that you can grow algae.